When Gregor Mendel made conclusions about inheritance, he did not notice examples in pea plants where gender of flowers made a difference. Gender does, however, affect inheritance in some cases. But gender is then difficult to talk about because different organisms establish the different mating types differently. So there is not a universal way of genetically establishing two different mating types. So for example, there could be something, not a nuclear chromosome, but a cytoplasmic factor, say in some protists, which determines whether an individual is of one mating type or another. It could be whether the egg is fertilized or not, which will determine gender, say in bees or ants. When we talk about sex chromosomes in, say, humans or fruit flies, it is the males which are the heterogametic gender, being XY, and the females which are the homogametic gender, being XX. And by that, I mean, if you are homogametic, that means that all of your gametes have the same sex chromosome. So in female mammals, for example, if the females are XX, then all of the ova possess an X chromosome, whereas if the males are XY, half the sperm carry an X chromosome, half the sperm carry a Y chromosome. So males are heterogametic. There's two different kinds of sperm, whereas the females are homogametic. There's one type of sex chromosome in ova. But in birds and butterflies, the opposite is true. It is the males which are the homogametic gender, and the females which are the heterogametic gender. There we refer to the chromosomes as being Z and W as opposed to XY, because the sex chromosomes then had different origins. So the males and females existed before there was the XY chromosomal system that may determine gender in live-bearing mammals, but gender was determined otherwise in other ways in other groups. In many reptiles, it is the temperature of the nest. So if you take a bunch of reptile eggs, like turtle eggs, and you take half and you heat them up, and you take the other half and you cool them down just a couple of degrees, that you end up getting, all males in one hatch and all females in the other hatch. And so temperature can play a role. In humans, as we will see, once again, as in all live-bearing mammals, with the egg-laying mammals, it's very confusing. There's multiple sex chromosomes involved. In the live-bearing mammals, the females are homogametic, putting an X chromosome in their ova. The males are heterogametic, and then it is then the Y chromosome which will determine the gender of the offspring. So Y-bearing or embryos fertilized by Y-bearing sperm are male chromosomally. Now, gender is much more complex, but that's not the purpose of this video, to talk about the gonadal stage of gender, the somatic stage of gender, etc. I'm just talking about the chromosomal aspect. Once again, there are multiple stages to gender, but that works itself into other courses. Now, I'll be referring to fruit flies in some of the courses, and it just is, fortunate for researchers that there is a similarity between fruit flies and live-bearing males, in that the females are homogametic, making what we call, the sex chromosome, or chromosome 1, whereas the males are heterogametic. So we could refer to XX females and XY males. These are not homologous to the X and Y chromosomes of mammals. And actually, it's a little bit more complex in flies. So in mammals, having a Y chromosome would make you male, chromosomally. Once again, there's other steps to gender. Whereas in flies, the answer is the ratio of Y chromosomes to the autosomes. And so if the XY system in flies was identical to that of humans, then this individual would be female because they don't have a Y chromosome. Whereas in flies, they are male. Whereas in humans, this individual would be male, whereas flies, they are female. So while the Y chromosome is involved in gender, it's the ratio of the chromosomes, which is what's going to absolutely determine gender, not the mere presence of a Y. So the first point I'm making here is that gender is established in different organisms differently. And so thus sex-linked inheritance would then be different in different organisms. So what I'm about to say applies to humans and live-bearing mammals, but wouldn't necessarily be universally true. So in humans, the 23rd pair of chromosomes, what are known as the sex chromosomes, have two alternate forms. There is the long form, which is known as the X chromosome. The reason it's known as the X chromosome is that after the S phase of the cell cycle, this is duplicated, but the centromeres are still joined. So if you have two of these long rod-shaped chromosomes, which are still attached in the middle, then it looks like an X. And so the females are XX, while the males have one long form of this chromosome, an X, and then a shorter form, which is the Y chromosome. And so, and this is significant because the Y chromosome, being much smaller, doesn't have as many genes. When females make their ova, while their cheek cells, their bone cells, their muscle cells have both of these X's in them as they are diploid cells. When they make ova, the ova will have this X chromosome or this one, but not both, with rare exceptions, which will be covered later. When males make sperm, the sperm are heterogametic. They either carry the X chromosome or the Y chromosome. If an X-bearing sperm fuses with an X-bearing ovum, which all of the ova are, then you would get an embryo which is now XX and female. And so if the sperm carries an X and the ova carries an X, then these individuals would be XX and female. But if, however, the sperm carries a Y chromosome, you have an embryo, and that's an individual who is XY and male. So this is how chromosomal gender in humans is determined. Once again, there are other steps in gender development, which are covered in different videos. So it is which sperm fertilizes the ova, which will determine the chromosomal gender of the offspring. And this is then why there's a form of inheritance known as sex-linked inheritance, because there are genes on these sex chromosomes, but far more on the X, which is about as long as chromosome 4, the fourth longest human chromosome, and much fewer genes on the short Y chromosome. If a female had a recessive mutation on her X chromosome, say for color blindness or muscular dystrophy or hemophilia, she would not then have the disease. She would not have hemophilia or color blindness, etc. And the reason is, if it's a recessive trait, then her other X chromosome would then presumably have the wild type dominant allele. And so she would have this and potentially pass it on to her children, but she would not have the disease. So she would be a carrier with the normal phenotype. Males cannot be carriers for sex-linked traits. They either have it or they don't, because if there is a recessive mutation on the X chromosome, the Y chromosome has so few genes that odds are it doesn't have a good copy of the gene to compensate for the mutation. So this male then would be affected with the genetic disorder. So males simply need one copy of a sex-linked mutation to express the trait and be affected. Women, however, would be two, which then means that Punnett squares get a little more complex. Now, one of the things I always recommend to my students is when you're making a Punnett square for a different a different trait, maybe a normal, dominant, or recessive trait. People pick whatever letter they want, maybe a C for cystic fibrosis, maybe a T for tongue rolling. But here, if you use those letters, you're going to get the question wrong. You need to use X's and Y's. And if you want to indicate the X chromosome that has a recessive mutation on it, maybe you put a dot at the top of the X. So here's a normal X, and here's like an X with a dot. So what would happen if a woman who was a carrier, she wouldn't have this sex-linked trait, let's just say it's color blindness. She isn't colorblind, but one of her two X chromosomes does have an allele for color blindness, and her partner is a male who is normal. What types of children would they have? Well, any children resulting from this sperm fusing with the ovum would then be XX and female. Any ones with this smaller Y chromosome would be XY and male. Half of the woman's ova are normal. So then the half of her children, which result from sperm fertilizing an ova with this chromosome, they would be completely normal. But then half of her children would then get an X chromosome with the mutation. Now, if a female receives an X chromosome with an allele for color blindness, this female would not have color blindness. Once again, she would have the normal phenotype, but would be a carrier who could pass it down to her offspring. However, this male, because the Y chromosome can't compensate for that, would then be affected. So the children of this couple would then be half, of the females would be normal phenotype non-carriers, half would be normal phenotype carriers, half the males would be normal, half the males would be affected. Remember, males cannot be carriers for a sex-linked trait. Here's another possible scenario. If the male is affected, if the male has a mutation on his X chromosome for, say, color blindness, then all of the children who receive an X chromosome from this man will then get that. Now, that means his daughters. His sons do not get an X chromosome from their father. The definition of being a son is to get a Y chromosome from your father chromosomally. And so all of the daughters receive an X chromosome with an allele for color blindness. If the woman does not have alleles for color blindness, then that means that these daughters would simply get one each, not two. So they would have normal phenotypes, not being carriers, not being colorblind, but they would be carriers, as opposed to the males, once again, none of which get that X chromosome from their father. So then they would simply be normal. In another scenario, if the male is affected and the woman is a carrier, then all of the daughters would get an X chromosome with a colorblind allele from their father. None of the males would get such a chromosome from their mother. Half of the ova would carry an X chromosome without an allele for color blindness, but then half would. So this would then be a carrier female. And then this individual would be an affected female. She would have gotten an allele for color blindness from both the sperm from her father and the ovum from her mother. So sex-linked traits can affect women, just to a lesser degree. The statistics I've seen are, almost 10% of American men are colorblind, but it's only about 1% of American women, because men only have to receive one X chromosome with that allele, while women have to receive it from both their father and mother. Half of the males would be normal and not have color blindness because they inherit this X chromosome, but half would then be colorblind because they would inherit that X chromosome. Okay, and so if you were describing the children of this this couple, you'd have to specify males and females. So half of the females are normal phenotype but carriers, half are affected with color blindness, half the males are normal, half the males are affected. You would not be simply saying 3-to-1 ratio the way you did with, say, a recessive disorder. Now, here you can obviously see the sex linkage aspect to this, because if a woman were affected, if she were colorblind, that means that all of her X chromosomes would then carry the allele for colorblindness. That means that all of her children would inherit that. If the male was normal, non-colorblind, that means that all of the daughters would be normal phenotype but carriers. All of the males would be affected. I'm sorry, there should be a red a star there as well. And so there are a number of disorders which are then sex-linked. All of these are on the X chromosome. Now, there are other disorders which are sex-influenced. The difference between, say, a sex-linked trait and a sex-influenced trait is that the sex-linked traits, the gene is on the X chromosome. And this then, sets up physiological differences, such as, different productions of hormones, et cetera. But now that there's different hormones, so if then males are typically making, say, more testosterone, less estrogen, females are making more estrogen, less testosterone, then those hormonal differences could then cause other differences later. So, for example, male pattern baldness or facial hair growth could then not be because there is a gene on the X chromosome, but because the chromosomal gender caused gonadal changes which affected the hormones made, and it's then the higher level of testosterone in this gender or the higher level of estrogen in that gender which is responsible for those other changes. And so in addition to some traits being sex linked, then there are other traits which are sex influenced or gender influenced because of hormonal differences, etc. And so, for example, let's say breast cancer. If you said had a male and a female with the same genotype in, say, their breast cells, the woman might develop cancer and the male might not. Why? Well, the woman makes estrogen with each menstrual cycle, which commands the cells to divide. Every time the cells divide, there are additional chances that more mutations would develop. And so, therefore, the cancer that this woman might experience is there was one maybe mistake that she was conceived with, but the frequent cell divisions caused by estrogen released during the menstrual cycle then was the stimulus for a second mutation, which then led to abnormal cell growth. This individual might have then had that first preliminary mutation, but without those frequent commands to divide, a second mutation did not develop. So this is sex-influenced, but not sex-linked. And notice that these genes are on chromosomes which are not the X and Y, because the males and females have equivalent chromosomes. So these are on autosomal chromosomes. One final comment is we often say that the gender ratio is, a 1-to-1 male to female ratio. That's not true. But it changes over time. At conception, there are far more males than there are females. There are perhaps 130 male conceptions for every 100 female conceptions. So there is a bias towards male conception in, once again, the live-bearing mammals. Why? Well, for a microscopic sperm, swimming the length of the vagina, the length of the uterus, and the length of the fallopian tube is quite a long journey. So For a microscopic cell, this is quite the journey. Now, sperm really don't have that much to them. They have a midpiece with some mitochondria, they have a flagellar tail, and then in their head, they have some enzymes and a cap. Other than that, it's just the 23 chromosomes. So to a large degree, sperm are chromosomes with a tail. Now, sperm which carry a Y chromosome are just a little bit lighter than sperm that carry an X chromosome because the Y chromosome is smaller. So of these sperm, some will, on average, swim just a little bit faster because they're a little bit lighter. Now, one wouldn't normally make a big deal out of that, but because sperm are so small, being essentially DNA with a tail, that a difference in the size of the chromosomal molecules, it makes a difference. And so it seems that this is why, on average, the sperm which reach the oocyte first are more likely to be Y-bearing sperm because they're just a little faster at swimming than the X-bearing sperm. So more males are conceived than females are conceived. But then if you were to wait until birth and ask now what is the gender ratio, the gender ratio is now about 108 male infants for every 100 female infants. So at birth, there are still more males than females, but the ratio was once 130 to 100. Now it is 108 to 100. What happened to 22 male embryos? And the answer is that they failed. Most miscarriages, most spontaneous abortions are male. Why? Well, the major reason is that if a female embryo has a mutation on the X chromosome, once again, it is a large chromosome with a lot of genes. If it's a recessive mutation, this female embryo is likely to survive, but then the male embryo would then be affected. And this very often causes a miscarriage, causes the male embryo to fail. So most miscarriages are male. But then at birth, there is still an excess of males, maybe 108 males for every 100 females born. By the time puberty is reached, the ratio is about 1 to 1. Well, then what happened to the infant males? If at birth there's 8 more males for every 200 compared to females, why is it a 1-to-1 ratio at puberty? And that is because on average, there are more infant deaths which are male than female. And once again, this is a major reason where males would then be affected by recessive mutations on the X chromosome, and then females would not. And so this video was meant to be part of a list of examples of exceptions or extensions to Mendelian inheritance. When Mendel described how traits get passed down from one generation to another, he did not notice traits being passed down differently to males or females. But it turns out they can be. But in talking about gender, there's so many other directions that one could go into. For example, how is gender determined in different organisms? That's a topic. In other videos, I will then discuss after chromosomal gender is established in humans, then how is gonadal gender established, or somatic gender, because some individuals, if you were to look at their chromosomes, you would predict that they would have this gender. But then when you looked at their gonads or their hormonal secretions, that is typical of the opposite gender. And so it is more complicated. But once again, the focus of this video was to give sex linkage as an exception or an extension to Mendelian inheritance, where gender matters in how traits are being passed down.